Our bodies are constantly bombarded by foreign invaders. These are called antigens. An antigen is any substance that can trigger an immune response. This could be a bacterium, a virus, a fungus, or even a non-living substance like pollen. When an antigen enters the body, it activates B cells, which then differentiate into plasma cells. Plasma cells are the antibody factories of our immune system. Antibodies, also known as immunoglobulins, are Y-shaped proteins. This unique shape is crucial to their function. The tips of the Y are called the variable regions. These regions are highly specific and can bind to a particular antigen. Antigens have specific sites on their surface called epitopes, or antigenic determinants. These are the specific parts of the antigen that antibodies recognize and bind to. The binding between an antibody and an antigen is highly specific, like a lock and key. The antibody's variable region must perfectly match the antigen's epitope for binding to occur. Once an antibody binds to an antigen, several things can happen. Neutralisation occurs when antibodies bind to a virus or bacteria, blocking its ability to infect cells. Agglutination happens when antibodies cross-link multiple antigens, forming large clumps that are easier for immune cells to clear. Opsonisation is when antibodies coat an antigen, making it more recognizable and palatable for phagocytes, which then engulf and destroy the antigen. Antibodies can also activate the complement system, a cascade of proteins that leads to the destruction of the antigen, often by creating pores in the cell membrane, called lysis. The principle of antibody-antigen binding is used in many real-life applications. Blood typing relies on antibodies that recognize specific antigens on red blood cells. Rapid tests, like those used for COVID-19, also depend on antibody-antigen interactions to detect the presence of specific antigens in a sample. Vaccines work by exposing the body to a weakened or inactive antigen, stimulating the production of antibodies and creating immunological memory. This allows the body to mount a rapid and effective response upon future exposure to the real antigen, granting immunity. Understanding antibody-antigen interactions is fundamental to grasping the workings of the immune system. From vaccines to diagnostic tests, this principle underpins many aspects of modern medicine. In educational settings, these interactions are often demonstrated through models and experiments. By visualizing how antibodies and antigens engage with each other, students gain a deeper understanding of immunology. Our lab focuses on developing new technologies that harness antibody-antigen interactions. These innovations could lead to breakthroughs in disease treatment and prevention. Using our knowledge of antibodies and antigens, we can better educate communities about the importance of vaccinations and regular health screenings. As we continue to explore and understand these critical interactions, we move one step closer to a future where diseases can be effectively managed or even eradicated entirely. Together, through science and understanding, we can achieve a healthier world for all.